Hi guys, this is Professor Beda again, and I will cover chapter 27, bacteria and archaea. As I mentioned before, prokaryotes are microorganisms. They are extremely small. They are teeny tiny, and they are microscopic. And even so, they are the dominant form of life on this planet. They are the most diverse set of organisms. Um, Prokaryotes, they are the first organisms to inhabit Earth, were prokaryotes that lived in 3.5 billion years ago. Most prokaryotes are unicellular, although the cells of some species remain attached to each other after cell division. That means they form colonies. And prokaryotic cells typically have a diameter of 0.5 to 5 micrometer, much smaller than that the 10 or to 100 micrometer of many eukaryotic cells. Prokaryotic cells have a variety of shapes. Cocci and singular coccus are spherical prokaryotes. They can occur singly or in chains of 2 or more cells, or they may occur in clusters resembling bunches of grapes. And we have bacilli, singular bacillus, are rod-shaped prokaryotes. They are usually solitary, but some forms, the rods are arranged in chains. We have spiral prokaryotes, include the spirochetes shown here, which are corkscrew-shaped. Other spiral prokaryotes resemble comas or loose coils. A key feature of nearly all prokaryotic cells is the cell wall, which maintains a cell shape, protects the cell, and prevents it from bursting in hypotonic solution or environment. The cell wall of prokaryotes differ in structure from those of eukaryotes. In eukaryotes that have cell wall, such as plants and fungi, the cell wall are usually made of cellulose or chitin. In contrast, most bacterial cell walls contain peptidoglycan, a network of sugar polymers cross-linked by a polypeptide. Archaeal cell walls contain a variety of polysaccharides and proteins, but lack peptidoglycan. Using a technique called the Gram stain, developed by the 19th century Danish physician Hans Christian Gram, scientists can categorize many bacterial species according to differences in cell wall composition. The structure of a bacterium's cell wall determines the staining responses. We have gram-positive bacteria have relatively simple wall composed of thick layer of peptidoglycan. The wall of gram-negative bacteria have less peptidoglycan and are structurally more complexes with an outer membrane that contains lipopolysaccharides. Lipid protein of lipopolysaccharides in cell wall of many gram-negative bacteria are toxic. Here we have the cell wall and plasma membrane of gram-positive and gram-negative bacteria. And this is the difference between them, and this is the gram staining to determine or distinguish between positive and negative gram bacteria. Gram-positive bacteria stain darker color and have a thick cell wall made of peptidoglycan, while gram-negative bacteria have a thinner layer of peptidoglycan located between the plasma membrane and outer membrane. plasma membrane and outer membrane. And you see with the blue or darker color, those are gram-positive bacteria. Gram-negative bacteria tend to be more resistant than gram-positive bacteria or species to antibiotics because the outer membrane impedes the entry of some drugs. The cell wall of many prokaryotes is surrounded by a thick layer of polysaccharide or protein. This layer is called the capsule, coated the outer cell wall. Some prokaryotes stick to their substrate or to one another by means of hair-like appendages called fimbriae, singular fimbria. Some bacteria uses fimbriae to fasten itself to the mucous membrane of its host. Fimbriae are usually shorter and more numerous than pili. Pili, singular pilus, appendages that pull two cells together prior to DNA transfers from one cell to the other. The cells of prokaryotes are simpler than those of eukaryotes in both their internal structure and the physical arrangement of their DNA. Prokaryotic cells lack the complex compartmentalization associated with the membrane-enclosed organelles found in eukaryotic cells. However, some prokaryotic cells don't have specialized membranes that perform metabolic functions. The membranes are usually enfoldings of the plasma membrane inside to the prokaryotes. The genome of prokaryotic cell is structurally different from eukaryotic genome and in most cases has considerably less DNA. Prokaryotic typically have one circular chromosomes, whereas eukaryotic usually have several to many linear chromosomes. Prokaryotes lack a nucleus. Their chromosome is located in nucleoid, and a region of cytoplasm, the nucleus, this dense region in the cytoplasm that is not enclosed by membrane. In addition to its singular or single chromosome, a typical prokaryotic cell may also have much smaller rings of independently replicating DNA molecules called plasmids, most carrying only a few genes. Alpha DNA replication, transcription, and translation are fundamentally similar processes in prokaryotic and eukaryotic cells. Some of the details differ. For example, prokaryotic cells or prokaryotic ribosomes are slightly smaller than eukaryotic ribosomes and differ in their protein and RNA content. These differences allow certain antibiotics such as erythromycin and tetracycline to bind to ribosomes and block protein synthesis in prokaryotic cells, but not in eukaryotes. As a result, people can use these antibiotics to kill or inhibit the growth of bacteria without harming themselves.